Imagine the explosive power of an Olympic sprinter just tearing down the track. You are watching the absolute peak of human biomechanics in action.
Oh, absolutely. It's incredible to see.
Right, and every single muscle is firing in this perfect synchronization, propelling them forward at incredible speeds. And then suddenly there's a sharp grab at the back of the thigh, a really devastating pop.
Yeah, the worst nightmare for a runner.
Exactly. The runner pulls up, limping, clutching their leg in agony, and their race is just instantly over. It is the dreaded pulled hamstring. And if you have ever wondered why this specific injury plagues so many athletes, and what is actually physically happening under the skin when that tissue fails, well, you are in the right place today.
We are definitely going to get into the weeds on this one.
We are, because to find out exactly why that happens, today we are exploring a 2024 paper from the Journal of Sports Science and Medicine. It's titled Fascicle Behavior and Muscle Activity of the Biceps Femoris Longhead During Running at Increasing Speeds. The research was authored by Lizhi Dahua, Shangjun Huang, Wu, and Mianfang Ruan.
And the mission for our deep dive today is to basically look directly under the skin of running athletes in real time. We are going to analyze in vivo data.
And in vivo meaning like live humans, right?
Right, exactly. Data gathered from living, moving human bodies. We're using this to fundamentally challenge a long-held myth about how human muscles stretch. By the end of our conversation, you will understand the true hidden mechanics behind the hamstring strain and see why the human body is far more complex than a simple rubber band.
I really want to start with those old assumptions, because to appreciate how groundbreaking this new data is, we kind of have to understand what scientists historically believed was happening when a hamstring tears.
You have to know what the baseline was?
Exactly. So let's ground this physically for a second. When we talk about a hamstring strain, where exactly are we looking on the body?
So we are focusing on a very specific muscle called the biceps femoris longhead. If you reach down to the back of your thigh, the hamstring is actually a group of muscles.
Okay, so it's not just one big muscle?
No, it's not. The biceps femoris longhead is that really thick, powerful band running down the outer portion of the back of your thigh. And it has this dual mechanical job. It works to extend your hip backward, and it also bends your knee.
Wow, so it's doing two different things across two joints.
Exactly. During a sprint, it is managing both of those extreme forces simultaneously.
And when athletes injure that specific muscle, they usually do it at top speed. So for decades, researchers must have been, trying to figure out what goes wrong at that maximum velocity.
They absolutely were. And for years, the scientific consensus was that these injuries were caused by something researchers called excessive strain.
Excessive strain, okay.
Yeah. The logic was fairly straightforward, really. The idea was that the muscle fibers were simply being stretched far beyond their physical limits until they snapped.
Which sounds totally logical.
Right. But the problem, however, was the methodology behind that consensus. For a long time, scientists couldn't actually look inside a sprinting human leg.
Because, how would you even do that?
Exactly. So they relied heavily on in vitro animal studies.
Wait, like dead animal muscles?
Yes, literally testing isolated dead animal muscles in a laboratory setting. And then they were taking that data and feeding it into static computer simulations to just guess what a living human leg was doing.
Okay, let's unpack this, because that seems like a massive leap of faith. You are taking isolated tissue, pulling on it in a lab, and then asking a computer to extrapolate that data to a human being running at, 25 miles per hour. What were those computer models actually predicting?
Well, the models were deeply focused on a fraction of a second in the running stride, known as the late swing phase.
The late swing phase. So what does that look like?
Picture a sprinter in slow motion. The late swing phase is that exact microsecond when your leg reaches out as far as it can in front of you. Your knee is extending and your foot is hovering just millimeters above the track, right about to strike the ground.
Oh, right, when the leg's completely outstretched.
Exactly. At that specific moment, your entire muscle tendon unit, which we call the MTU, so that's the entire system of the muscle belly and the tendons attaching it to the bone, that entire unit is lengthening to accommodate the stride.
Makes sense. It has to reach out.
Right. Well, the computer simulations mathematically predicted that the actual muscle fibers inside that unit, the fascicles, were stretching up to 40 millimeters.
Wait, 40 millimeters? So almost 2 inches of physical stretch in the muscle fibers themselves.
Yeah, that was the prediction.
That paints a very specific picture. So the old theory is essentially treating the muscle like a giant rubber band. You swing the leg forward, the rubber band stretches out, and if the athlete reaches too far, the rubber band stretches past its capacity and snaps. That's the excessive strain.
That was totally the prevailing mental model. According to those old computer calculations, the muscle fibers were taking on about 80% of the total stretch of the entire leg extension.
80%?
Yeah, the assumption was that the muscle itself was doing almost all the expanding. But a computer simulation of a rubber band doesn't account for the complex neurology and structural dynamics of a living human.
But wait, computer models and animal muscles in a lab are one thing. Is the human leg really just a simple rubber band in motion? I mean, which naturally leads to a massive problem for sports scientists.
Oh, absolutely.
If you suspect the computer model is wrong, how do you prove it? Because you can't exactly put an athlete in an MRI machine and tell them to sprint at full speed. So how did the authors of this 2024 paper actually manage to measure muscle fibers inside a moving human being?
Well, they had to pioneer a deeply challenging experimental setup. They brought in 7 male college sprinters.
Yeah.
And these are not casual joggers, right?
Right.
These are serious, highly trained athletes with personal bests around 11.1 seconds in the 100-meter dash. The researchers put them on a highly specialized instrumented treadmill and had them run at speeds of 4, 5, and 6 meters per second.
Wow, 6 meters per second is really moving. That is a very aggressive pace for a treadmill.
It is.
I'm trying to visualize how they captured data from inside the leg at that speed without, completely interfering with the runner's stride.
The technology application is honestly a marvel of biomechanics. They utilized standard 3D motion tracking cameras to capture the skeletal movement and force plates in the treadmill to measure the exact impact of the foot.
Standard lab stuff, right?
Right. But the breakthrough was the ultrasound. They took a 130-gram, 7 megahertz ultrasound probe and physically strapped it directly to the back of the sprinter's thighs using these specialized compression bandages.
Wait, just to be clear, they bound a medical ultrasound machine to the leg of a guy sprinting on a treadmill.
They did, yes. And alongside that probe, they attached EMG sensors, so electromyography sensors, to the skin to measure the raw electrical activation of the nervous system firing into the muscle.
So they're tracking everything at once.
Exactly. The goal was to simultaneously track the mechanical movement of the tissue and the electrical command coming from the brain.
But how does an ultrasound even track a muscle fiber during a violent movement like a sprint? I mean, I am used to seeing ultrasounds in quiet, still hospital rooms, not bouncing around on a treadmill.
Well, what's fascinating here is that the probe sends high-frequency sound waves into the leg, and those sound waves bounce back when they hit different tissue densities. The researchers were specifically using it to track the deep aponeurosis and the superficial aponeurosis.
Okay, let's define aponeurosis for a second, just so we want everyone to have a really clear mental image of what is being tracked.
Sure. Think of an aponeurosis like the tough, silvery, fibrous casing that sheathes the muscle. It is connective tissue.
Oh, like that silver skin you see on a piece of steak.
Yeah, that's actually a great analogy. So by tracking the distance and angle between the superficial casing on top and the deep casing underneath, the ultrasound can actively watch the muscle fascicles, the actual fibers housed between those layers, change length.
And it does this while the person is sprinting.
Yes. And this probe was capturing that movement at 22 frames per second while the athlete was in motion.
That is insane. So we are no longer looking at an isolated animal muscle on a lab table or a theoretical equation in a computer. We have highly trained athletes wired up with motion capture dots, electrical sensors, and an ultrasound probe, giving us a live video feed of the muscle tissue tearing away on this treadmill.
Exactly. Live in vivo data.
So if the computer models predicted a massive 40-millimeter stretch in the muscle fibers during that late swing phase, I have to imagine the ultrasound showed those fibers stretching to their absolute tearing point. What did they find?
Well, the live data showed the exact opposite. Yeah, it completely dismantled the old simulation models. So during that late swing phase, the entire muscle and tendon unit, the MTU, did indeed lengthen significantly. The leg is reaching out in front of the body, so the anatomical system from the hip to the knee has to physically get longer.
Obviously, yeah. The leg is stretching out.
But when the researchers looked at the ultrasound feed of the actual muscle fascicles, the fibers themselves barely stretched at all.
Wait, barely stretched. We're talking about maximum leg extension during an explosive sprint. How much did the ultrasound actually show them lengthening?
Only about 1 centimeter. 10
Are
millimeters.
you serious? That is an unbelievable discrepancy.
It really is.
The accepted scientific models predicted 40 millimeters of stretch, and the reality inside the living leg was 10 millimeters. The computers were off by a factor of 4.
Yeah, the contrast fundamentally changes our understanding of the anatomy. Instead of the muscle fascicles doing 80% of the stretching, the live ultrasound data proved that the fascicle lengthening accounted for only about 20% to 32% of the total length change of the entire leg system.
But if the entire leg is definitely stretching out to take the stride, and we now know the actual muscle fibers are barely moving, where is all that length coming from? I mean, something has to be spanning that physical distance.
The length is coming from the connective tissues. The tendons are taking over.
Oh, the tendons.
Yeah, they are stretching and doing the heavy mechanical work of lengthening. Meanwhile, the actual muscle fibers stay relatively static. They are performing what biomechanists call a quasi-isometric contraction.
Meaning they're basically holding still.
Exactly, they're holding their length.
Right.
This physiological phenomenon is known as decoupling.
Decoupling.
Right. The physical behavior of the muscle fiber is entirely decoupled or disconnected from the behavior of the overall muscle tendon unit.
Okay, let's build a visual for this decoupling concept for the listener, because the old rubber band analogy is completely dead now.
It's totally dead.
If the tendon is doing all the stretching, the tendon is acting like a bungee cord. It gets pulled and expands, but what is the muscle doing? If it's performing a quasi-isometric contraction, meaning it's contracting to stay the exact same length, well, it's not a bungee cord at all.
No, think of the muscle more like a rigid metal chain trying to hold its links tightly together.
Oh, a metal chain. So instead of a simple rubber band, it's more like a rigid metal chain attached to a bungee cord.
That is a perfect way to visualize it.
When the runner kicks their leg forward, they pull the whole system. The entire thing gets longer, but it's the bungee cord, the tendon taking all the stretch, absorbing that energy, while the metal chain, the muscle fiber, is locking down, staying completely rigid.
Exactly. The tendon stretches to store elastic energy, which it will eventually use to snap the leg back down to the ground. The muscle fibers deliberately tense up and hold their length to protect themselves from tearing and to transfer that massive force efficiently.
Wow, this decoupling phenomenon is fascinating, but it brings up a glaring contradiction.
What's that?
Well, if the muscle fibers aren't actually overstretching, if they're locking down like a metal chain and only moving a single centimeter, then why in the world do hamstrings still tear during sprints? If there's no excessive strain snapping the rubber band, what is causing the athlete to grab the back of their leg in agony?
To solve that mystery, the researchers had to look away from the ultrasound and look at the EMG data. They had to look at the electrical signals coming from the nervous system.
The EMG data.
Right. The ultrasound showed us the mechanics, but the EMG showed us the sheer neurological violence of the sprint.
And the numbers from that electrical data were staggering. I remember the paper noted that when the sprinters hit 6 meters per second, the electrical activation of the hamstring muscle peaked at 116% of maximum voluntary contraction, or MVC.
Yes, 116%.
Wait, how can a muscle fire at 116% of its maximum? Isn't maximum by definition 100%? It sounds mathematically impossible.
It sounds like a paradox, I know, but it comes down to how baseline measurements are established in sports science.
Okay, how so?
Before the runners ever got on the treadmill, the researchers established their 100% maximum voluntary contraction baseline using a static isometric test. They had the athlete lie face down on a table, bend their knee against a padded resistance arm, and consciously push as hard as they possibly could.
So it's a completely conscious effort.
Exactly. Whatever force they generated purely through their own willpower became the 100% baseline.
So 100% is simply the hardest your conscious brain can tell your muscle to push while lying safely on a clinical table.
Yes, but dynamic, explosive running is an entirely different neurological state. When you are sprinting, you're not just using conscious willpower. Your central nervous system triggers automatic, reflexive motor patterns designed for high-speed survival and locomotion.
That makes sense. It's almost primal.
It is. In that dynamic state, the nervous system can bypass conscious limiters and recruit additional motor units, literally more muscle fibers than you could ever possibly activate just by thinking about it on a table.
So sprinting literally unlocks a hidden gear in your nervous system. The brain sends a signal so powerful it overdrives the muscle to 116% of its conscious capacity. But why is the brain sending such an overwhelmingly powerful signal right at that specific moment?
Well, if we connect this to the bigger picture, the timing is the ultimate smoking gun for why the hamstring tears.
Okay.
That 116% electrical spike perfectly coincides with the peak length of the leg extension, right at about 90% of the sprinting gait cycle.
Just right before the foot hits the ground.
Exactly. The leg is swinging forward at maximum velocity, the bungee cord is fully stretched, and right before the foot hits the ground, the nervous system blasts the hamstring with over 100% of its maximum electrical power to hit the brakes.
Wow, it's an emergency brake. The hamstring has to violently contract to slow down the swinging lower leg. Otherwise, the momentum would literally rip the knee joint apart.
It absolutely would.
So it stops the forward swing and instantly prepares to pull the leg backward the microsecond the foot strikes the track.
Imagine the sheer physics involved here. The lower leg is still traveling forward with immense momentum, but the hamstring muscle is contracting with violent electrical force in the opposite direction.
It's like a tug of war.
Yes, it is trying to lock down and act like that rigid metal chain, fighting against the forward momentum of the bone. This creates an unimaginable amount of tensile stress.
Stress. Okay, that is the key word to redefine here. We talked earlier about the old theory being excessive strain.
Right. Strain refers to an object stretching too far until it breaks. The old theory assumed the hamstring tore because of length, but the new data shows it is a failure of force.
It's about force.
The injury isn't caused by the fibers stretching too far, so not strain. It's caused by the sheer mechanical force and tension stress ripping the tissue. The muscle is fighting so hard to maintain that quasi-isometric lockdown that the sheer physical tension of the opposing forces literally rips the tissue apart from the inside.
That is wild. So it is not about a lack of flexibility. It has nothing to do with not stretching enough before a race. It is entirely about the muscle's structural ability to withstand mind-boggling amounts of tension while trying to hold perfectly still against the momentum of a sprinting leg.
Exactly. That is a total paradigm shift in biomechanics. It changes everything about how we view the kinetic chain of running.
It really does. But, as with any rigorous scientific exploration, we have to recognize the boundaries of this specific experiment.
Absolutely.
The authors were incredibly transparent about the limitations of their work, and we need to look at those to understand how this translates to the real world.
Right, and first off, there's the speed. The runners in this study hit 6 meters per second. Now, for an average person, that is an all-out sprint. But for these elite collegiate athletes, 6 meters per second is actually only about 70% of their absolute maximum speed.
That is a crucial caveat. We're operating on the highly educated assumption that these decoupling mechanics hold true at 100% maximum Olympic speed.
Right, but we don't have the live ultrasound data for a human running 10 meters per second yet. Strapping equipment to a leg at that velocity risks altering the runner's natural biomechanics, or simply tearing the equipment right off the body.
Yeah, the equipment would just fly off. And there is also a physical limitation with the ultrasound probe itself, right? The probe they used was 4 centimeters long. But a human hamstring fascicle is much longer than 4 centimeters.
Exactly. Because the entire muscle fiber couldn't fit in the viewing window of the 4-centimeter probe, the researchers had to use mathematical trigonometry to extrapolate the full length of the fascicle based on the angles they could see.
It's an estimation.
Right. It is a highly validated and standard method in biomechanics, but it is technically an estimation, not a full end-to-end measurement.
Got it. And the final limitation is perhaps the most relevant to why injuries actually happen in competition. These runners were fresh, they were tested in a controlled environment, completely unfatigued.
And that changes the physiological environment entirely. But, even with those limitations, this deep dive into the true mechanics of stress versus strain fundamentally alters how we should approach athletic training.
So what does this all mean for you? Whether you are a competitive athlete, a coach designing a training program, or just someone who occasionally sprints for the bus. If we now know that hamstring tears are caused by massive tension under a quasi-isometric lockdown and not by stretching too far, how does that change what we do in the gym?
Well, it means that simply sitting on the turf and doing static toe touches is not going to protect your hamstring from tearing during a full sprint.
Flexibility isn't enough.
No.
Oh.
If the injury is a failure to handle high stress while the leg is extended, your rehabilitation and injury prevention should focus on eccentric strength training.
Okay, let's define eccentric for the listener. Because we know isometric means the muscle is holding still. Concentric means the muscle is shortening, like curling a dumbbell up. But eccentric means the muscle is actively contracting and generating force, but it is being forced to lengthen at the same time.
Right. And the gold standard example for this specific injury is the Nordic hamstring curl.
Oh, those are brutal.
They are. You kneel on a padded floor, a partner holds your ankles firmly to the ground, and you slowly lower your rigid torso forward toward the floor. using only your hamstrings to act as the brakes against gravity.
And as you fall forward, those hamstrings are firing as hard as they can, but the weight of your body is slowly forcing them to elongate.
Exactly. What this specific exercise is doing neurologically and structurally is training the muscle fibers to handle massive amounts of tension while acting quasi-symmetrically.
It's mimicking the sprint.
You are literally rehearsing the precise mechanical demand that a sprint will eventually place on the muscle. You are training the metal chain to be stronger, denser, and more resilient, so it doesn't snap under the immense stress of a sudden braking force.
It makes perfect sense. You are systematically exposing the tissue to high stress in a safe, controlled environment, so it survives the chaotic, high-stress environment of a race. It is amazing how looking past the computer models and actually looking under the skin changes our entire philosophy on movement. We thought we needed more flexibility, but what we actually needed was a stronger structural chain to handle the tension.
It really highlights why in vivo data is so vital to pushing science forward. But, I keep coming back to that final limitation we discussed.
The fatigue factor.
Yeah, the fact that the athletes in this study were completely fresh and unfatigued. It leaves us with a fascinating, critical question that researchers still need to answer.
Which is what?
Well, if our tendons are acting like bungee cords while our muscle fibers rigidly lock down to protect themselves, what happens when we get fatigued?
Oh, wow.
As the race goes on and the muscle tires, does that protective neurological lockdown suddenly fail? And is that split-second failure why injuries usually happen at the end of a sprint?
That is an incredible, provocative thought to end on, and a perfect setup for whatever research comes next in this field. This has been a scientifically rigorous look at the hidden mechanics of human running. We highly encourage you to read the full article by Mao and colleagues to explore their complete methodology and data. Also, remind yourself to follow the channel and turn on notifications for similar content. The next time you watch a sprinter tear down the track, you won't just be watching their speed. You will be picturing the metal chains, the bungee cords, and the immense hidden forces keeping them perfectly in stride until, of course, they fail.